Your kids could get free or low-cost health coverage from Medicaid or CHIP. Even if you've applied before, they may be eligible now. Kids up to age 19 are covered for checkups, vaccines, dentist visits, hospital care, and more. And if they already have Medicaid or CHIP, remember to renew every year. Visit insurekidsnow.gov or call 877-KIDS-NOW. Paid for by the U.S. Department of Health and Human Services.
Hey everyone, welcome back to my podcast, Anatomy and Physiology Bit by Bit. This is your host, Dr. Steve Sullivan, coming to you from Bucks County, Pennsylvania. So I'm glad to have you back. We are now about ready to enter into the circulatory system. I'm excited for that because that is one of my favorite systems to teach about. So I'm very excited to get into this and finally get out of that endocrine system. That was a lot of episodes. But before we jump into circulatory, I do want to address a question that I got through email. So this question comes from Liam, and Liam is writing to me from Louisville, Kentucky. And that is in the United States, for those of you who are listening from outside the US. And his question is about endocrine disruptors. And I didn't cover anything about that in the endocrine system, but that's a really good question because what are they and what do they do? And that's basically what Liam's question was what are endocrine disruptors and should we be worried about them? And so I think this is a good question to address as we open up this system and leave the endocrine system, but with one little quick tag about endocrine disruptors. So what I want to mention is that, remember that the endocrine system is all based on hormones, right? So we've got all these hormones, hormone receptors, hormone synthesizers, glands, the bloodstream, target cells, all those things, all related to hormones and their effects and how they have their effects. So a hormone disruptor or an endocrine disruptor is any man-made chemical that can get in the way of one of those systems or pathways. So, for example, if there is a chemical in something in the environment that gets in the way of insulin receptors, then it's going to make it difficult for your target cells to respond to insulin that's been released. And that is going to mean that it's a disruptor. It has disrupted the pathway of insulin. Now, that's one example, right? Endocrine disrupting chemicals, or EDCs, can mimic a hormone, they can block a hormone, they can interfere with the hormone's ability to get to its target cell. Any way along that pathway of the hormone being released, and then the hormone actually getting to its target cell and affecting its target cell can be disrupted. So now these are man-made chemicals, and according to the National Institutes of Environmental Health Sciences in the United States, there's about 85,000 human-made chemicals in the world. And about 1,000 of those or more could be endocrine disruptors. And that's based on the unique properties of the chemical itself. So there are a few types of hormone disruptors or endocrine disrupting chemicals that are known and that we're concerned about. So for example, atrazine. Atrazine is found in herbicides, which is like weed killers. So that is possibly an endocrine disruptor. Bisphenol A, or BPA, which is used to make some kinds of plastics, that can get in there. So a lot of times you'll see plastic containers or water bottles that say they're BPA free because you don't want that chemical leaching into the water or the food from the packaging or the bottle and getting into your body. Another one is dioxins. These are typically like a byproduct of certain manufacturing processes, like when you make herbicide or weed killers or something to bleach paper. And they can be released into the air from burning waste and also even from wildfires. Perchlorate is a colorless salt manufactured and used as an industrial chemical to make rockets, explosives, and fireworks. And what happens is it could be leached into the groundwater. So it can get under the ground, and then it starts to leach down through the soil into the groundwater and then it's going to get into our water system. There's a bunch of things in here. Phthalates, which is spelled Phthalates. That is a large group of compounds used as liquid plasticizers. So you're going to find a lot of these things in food packaging, cosmetics, fragrances, children's toys, medical device tubing, things like that, shampoo, cleanser, aftershave lotion. So those kinds of cosmetics, you want to try to check the ingredients and find out. There's a longer list here that I'm looking at through the National Institutes of Environmental Health Sciences, but you can read about it yourself. Basically, that's what I did. I looked it up because I'm not a big expert on endocrine disrupting chemicals, but I looked it up, and I'll put the link to the site that I found on the show notes so that you can look it up as well if you're curious about these endocrine disrupting chemicals. But now that you've listened to all the podcast episodes I have on the endocrine system, you'll have a pretty good idea of how hormones work. And that way, when you see how something gets in the way, how that can disrupt the hormone's function. And that's the problem, right? Because we know that these hormones are utilized for almost everything your body does. So anytime you disrupt them, you're going to get in the way of something important in your body. So that's an important thing to look at. So I'll put that website on the show notes, and hopefully you'll get a chance to look it up if you're interested. But thank you, Liam, from Louisville, Kentucky, for asking me that question. You can also ask me a question. If you want, just email me at minus55media@gmail.com, and I will try to answer your question on the show. All right, having said that, I think it's time to get into the circulatory system. And we're going to start with just an overview of the circulatory system. Because when we think about the circulatory system, it is 3 parts: the blood vessels, and the heart. So I typically split that up when I teach A&P. So this is going to be a long list of shows because we're going to go through the circulatory system, and typically it's 3 chapters in a 2-semester anatomy and physiology book. So we're going to cover it: blood vessels, and then heart in that order. So it's going to take a few episodes to do that. The circulatory system is the main system of transport in our bodies. And from studying the endocrine system, we have a pretty decent idea about that already, because that's how hormones get from their gland that synthesizes them and releases them to their target cells. But it's not just hormones. We're also sending nutrients, electrolytes, oxygen, and other materials that the cells need, as well as allowing cells to dispose of their waste products like carbon dioxide and urea. So this is a transport system not only for nutrients like glucose, but also for waste products like carbon dioxide. I'm going to start off with general features and functions of the circulatory system before we specifically get deep into blood itself. But speaking of blood is a fluid connective tissue that is composed of cells and a watery extracellular matrix. So if you remember way back from our episodes on histology, we talked about connective tissue, and connective tissue is defined as cells amidst an extracellular matrix. And blood is no different, but the extracellular matrix is 92% water, and that's what we call our blood plasma. And the cells are red blood cells, white blood cells, and cell fragments called platelets. Red blood cells we also call erythrocytes, and white blood cells we call leukocytes. And there's many different types of leukocytes, and we're going to get into them in detail. Now, blood is pumped into a series of tubes called blood vessels by a muscular pump called the heart. And those are the components: the blood, the heart, and the vessels. Now, certain microscopic vessels called capillaries allow the blood plasma and many of its solutes to be filtered out of the circulatory system and into the interstitial fluid surrounding our cells. Conversely, interstitial fluid and its solutes are reabsorbed back into the capillary and returned to the blood plasma. The blood circulates through these vessels, eventually returning to the heart, where it's pumped through again. And that's why we call it a circulatory system, because it's really kind of a circle. We can say we start at the heart, but we don't really start anywhere. But let's just say we're starting at the heart. The heart is going to pump blood and force it into the vessels with a certain degree of pressure. And that pressure will serve as the force that drives the blood forward, whereas the walls of the vessels serve as the resistance to that flow, making sure that the blood goes in the direction we want it. Now, when that blood eventually gets to these tiny microscopic capillaries, the force of the pressure will make it so the blood plasma leaves through tiny holes in those capillaries, which we'll get into in detail later, and taking with it whatever is dissolved in that blood plasma, like nutrients, chemical messengers, electrolytes, etc. And also, oxygen will diffuse from the red blood cells through the epithelial cells of the capillary and into the surrounding tissues. So that way, the cells that blood vessel is going to are going to get oxygen so that they can make their ATP. Now, when they make their ATP, they're going to produce as a byproduct, among other things, carbon dioxide. That carbon dioxide will then diffuse back into the capillary, along with interstitial fluid with its solutes, which could be waste products or cellular products that we need to get through the circulatory system, like hormones, for example. And then those capillaries, they lead back to a different kind of vessel called a vein, and that vein will eventually meet up with other veins and bring the blood back to the heart so we can start all over again. All right, let's talk about the composition of blood. Like I said before, blood is a combination of cells and an extracellular matrix, which is called blood plasma. And that blood plasma is about 92% water. And the rest of it is the solutes that are dissolved in the blood plasma. On average, a healthy adult human has about 4 to 6 liters of blood in the circulatory system. That's about 8.5 to 12.5 pints. The cellular component of blood consists of erythrocytes, leukocytes, and platelets. Now, erythrocytes are also known as red blood cells, and leukocytes are also known as white blood cells. Platelets are fragments of bone marrow cells, and they're also known as thrombocytes. These cells and the cell fragments are collectively known as the formed elements of the blood. When you hear the term whole blood, people are talking about the cellular component and the blood plasma together. So you might hear that term, especially clinically, whole blood. That's all of it, the cells and the plasma. The percentage of whole blood that is made up of the erythrocytes is called the hematocrit. It's measured by collecting a small amount of blood in a tube, which is then placed into a centrifuge, which spins it until the centrifugal force pushes all the formed elements to one end of the tube. The erythrocytes move to the bottom of the tube. The leukocytes and platelets are next in a thin line called the buffy coat, and the rest is the plasma. We can then measure what percentage of the whole blood consists of red blood cells by dividing its length by the length of the tube containing all the blood components. And that measurement is called the hematocrit. Typically, it's somewhere between 40 and 55%. By far, the most numerous of the formed elements is erythrocytes. Each microliter of blood has between 4.2 and 5.6 million erythrocytes for biological females and 4.6 and 6.2 million for biological males. If we compare that to leukocytes, you're going to see a massive difference because leukocytes only range somewhere between 5 and 10,000 per microliter. That means that there is up to 1,000 times more erythrocytes in our bodies than there are leukocytes. There's also about 130,000 to 360,000 platelets per microliter of blood. The erythrocytes are described as biconcave discoid cells with no nucleus or organelles. About a third of the cytoplasm of a red blood cell is made up of a red, pigmented protein called hemoglobin. Hemoglobin is a combination of 4 protein chains called globins, each bound to a non-protein heme group. In the center of each heme group is one iron ion that combined one oxygen molecule, meaning each molecule of hemoglobin has the ability to bind a total of 4 oxygen molecules and transport them throughout the circulatory system to be delivered to the tissues. Hemoglobin's main function is to carry oxygen, but it does carry a little bit of carbon dioxide as well. The red color of hemoglobin is what gives blood its red color. Despite the persistent myth that human blood is blue inside our bodies, human blood is always red, inside or out. Oxygen-rich blood is a little brighter red than oxygen-poor blood, but it's always red. If you hold a white light flashlight up to your hand and see what color shines through on the other side, you're shining that light through all the red blood coursing through the vessels in your hand. So of course, the color that comes through your hand is red. A lot of people believe that blood is blue when it's inside our bodies, and it becomes red when it is outside our bodies. That is not true. Blood is never blue, at least not human blood. If it was, when you shine that light, you would see blue coming through. Sometimes people think that because when they look at the veins through their skin, they look blue. That's not because of the fluid that's inside. That's because the wavelength light of blue is what's coming through your skin from those vessels. Since erythrocytes don't have mitochondria, they can only synthesize ATP via anaerobic fermentation, which does not utilize oxygen. Now you would think, why wouldn't they do aerobic respiration since they've got all that oxygen there? Well, one, they don't have mitochondria, so they can't do it. But two, if they did have mitochondria, the mitochondria would end up using up all the oxygen that the blood cells are trying to deliver to your tissues. It would be kind of like a gasoline truck transporting gasoline from one point to another, but using its cargo as its own fuel. By the time it got to its destination, there wouldn't be much left. So we don't want the red blood cells to use up all the oxygen that they're trying to deliver to your tissues, so they don't have mitochondria. Now, since they also don't have a nucleus, that means no cell division. No mitosis if there's no nucleus. So erythrocytes have a lifespan of about 120 days, and they die off. And we have to make new ones constantly to make sure we have enough. So they're not like skin cells or liver cells that are going to have mitosis and they're going to replicate their DNA and make 2 new daughter cells. Can't do that in erythrocytes. So we have to constantly make new erythrocytes through a process called erythropoiesis, which takes place in the red bone marrow of our bones. Now that 120-day lifespan means we also have to dispose of old, dead red blood cells, or old red blood cells that aren't functioning anymore. And our liver is taking care of most of that. Those cells break down, and then we have all these pigments inside that we have to dispose of. One of those pigments is called bilirubin. And if the liver has a hard time disposing of bilirubin, meaning that the liver's not functioning at 100%, so bilirubin ends up not being broken down by the liver and starts getting deposited into your skin and the whites of your eyes, called the sclera. And that will create a yellowish hue to those areas, and we call that sign jaundice. So the reason why jaundice is a sign of liver function decline is because that bilirubin should be getting disposed of. Now, one of the things that will help break down bilirubin is ultraviolet light. So one of the treatments for jaundice, if jaundice is not really a sign of liver failure, is ultraviolet light. So sometimes babies are born and their liver is not quite ready yet to break down its own bilirubin, and so the infant has jaundice. And it doesn't usually last all that long, but one of the treatments for that is this blanket that plugs into a machine, and the blanket has light sources all inside of it with ultraviolet light. And the baby will sit there with that blanket on, with the light, most of the day and night, and that will help break down the excess bilirubin that's building up so it doesn't cause any problems, until eventually the liver is ready to work it out on its own.
You buy a pair of socks, that's 2 socks. You buy a pair of Bombas socks, that's 4 socks. Because one purchased is one donated. Socks are the #1 most requested clothing item in homeless shelters. So when you buy a pair of super comfortable Bombas socks, you're also donating a pair. Bombas customers have powered over 150 million donations. So Bombas would like to thank you 150 million times. But we only have like 30 seconds. Go to bombas.com and use code AUDIO for 20% off your first purchase. That's BOMBAS.com and use code AUDIO at checkout.
Your kids could get free or low-cost health coverage from Medicaid or CHIP. Even if you've applied before, they may be eligible now. Kids up to age 19 are covered for checkups, vaccines, dentist visits, hospital care, and more. And if they already have Medicaid or CHIP, remember to renew every year. Visit insurekidsnow.gov or call 877-KIDS-NOW. Paid for by the U.S. Department of Health and Human Services.
So the process of erythropoiesis, let's break that down for a second. First, we have what's called a hemopoietic stem cell. And that is the first cell that's going to start to become a future red blood cell. And what that will do is that will give rise to an erythrocyte colony forming unit, or ECFU. The hormone erythropoietin from the kidneys and the liver stimulates receptors on that ECFU and causes it to differentiate into an erythroblast. That erythroblast begins multiplying into several erythroblasts that each engage in hemoglobin synthesis. Eventually, the nucleus shrinks and is shed from the cell, which is now considered an immature erythrocyte called a reticulocyte. Reticulocytes, which are now anuclear, still contain some ribosomes, but are deposited into the circulatory system by the bone marrow. The ribosomes are going to eventually disintegrate and disappear as each reticulocyte becomes a mature circulating erythrocyte. So if that question is, how do we make these cells with no nucleus? Well, we really don't. What happens is the nucleus is shed from the cell as it begins to shrink. So the cell gets rid of its nucleus rather than a cell forming from no nucleus, which we know can happen. Since we're on red blood cells, let's finish up this episode talking about blood type. And we'll get into white blood cells next episode. So I'll bet you've heard the term blood type before. I mean, you may even know your own blood type. But what you may not know is that blood type refers to the erythrocytes' surface antigens that you inherit from your parents, their parents, and so on. There are over 30 different groups of erythrocyte surface antigens, but we're going to focus on the 2 that are the most clinically significant and the 2 that you've probably talked about in the past. So I think before we get started talking about surface antigens, we should take a second to understand what an antigen is. Antigens are membrane proteins, glycoproteins, or glycolipids that are present on all cells. The antigens on an organism's cells are synthesized based on what that organism's genetic material is. And we know that genetic material, or DNA, is unique from individual to individual. We see antigens on animal cells, plant cells, bacteria, viruses, you name it. Sometimes you'll hear the term self-antigens because our immune system uses our own antigens to recognize our own cells. This way, your immune cells focus on attacking invading organisms and viruses, not mistakenly attacking your own. One of the tools our immune system uses is a class of proteins called antibodies, which are synthesized by specific white blood cells called plasma cells. Antibodies are usually specifically designed to bind an antigen so they and the cells they're attached to can be identified and rendered harmless. This is the reason most people are not infected by most viruses twice. We talked about this before with the common cold. Once the body develops antibodies against the virus's antigens, it can render the virus harmless the next time it infects the same body, as long as the virus hasn't mutated enough to change its antigens. And as long as you're still making enough of those antibodies to fight that virus. The first group of blood types we'll discuss is the ABO group. This group focuses on whether or not the plasma membrane of a person's erythrocytes contains one or both of 2 similar antigens. These antigens are called antigen A and antigen B. If the red blood cell expresses antigen A, it's considered to be type A blood. If it contains antigen B, it's type B blood. And that's pretty simple, right? Some people express both antigens, so we call that type AB blood. But then, of course, there's some people that don't express either of those antigens, and we call that type O blood. So I know it doesn't mean it, but I like to think of that as type 0, meaning which antigens do they have? 0. So type O blood means they don't express either of those antigens. Now, there's more to these types than the antigens, though. We have to be careful about mixing these blood types, because when we're babies, we start to produce antibodies in response to the bacteria that live in our digestive tract. And those antibodies make their way into the blood plasma. Now, some of those antibodies actually have an affinity for the antigens A and B on human red blood cells. The one that binds to antigen A is called the anti-A antibody, and the one that binds to antigen B is called the anti-B antibody. Now, under normal circumstances, we're never going to make antibodies against our own antigens. So people with type A blood have the anti-B antibodies in their blood plasma, and people with type B blood have the anti-A antibodies in their blood plasma. So remember, the bacteria that live in your digestive system, they're expressing antigens, and your immune system is making antibodies against those antigens. But those antigens are very similar to the A and B antigens we have on red blood cells. So those antibodies actually have an affinity for those antigens. Now, if you're type A and you have the A antigen, no matter what kind of bacteria you have in your body, you're not going to make an antibody that goes and attacks your own cells. If you did, that would be called an autoimmune disorder. We don't want that. So we're going to only make the one that attacks the B antigens, and we call those the anti-B antibodies. So if you are type A, you also have the anti-B antibody. If you are type B, you also have the anti-A antibody. Now, if you are type AB, you have both antigens, so you're not going to make any of those antibodies. So you have 0. And if you're type O, you have both antibodies because you have neither antigen. So what this means is that we have to be really careful when a patient needs a blood transfusion. If a type A person receives type B blood, that patient's antibodies will bind to the antigen B found on the donor blood's erythrocytes. One antibody can bind to multiple antigens on multiple cells, causing them to stick together and form large clumps of red blood cells in the blood vessel. That process is called agglutination, and those large clumps can clog the blood vessels and damage organs, which is often fatal. Now, there's more to transfusion compatibility than just the ABO group. But for a moment, let's consider some transfusion scenarios for just ABO group alone. If the host is type A, meaning the recipient of the blood transfusion is a type A blood person, that means it has anti-B antibodies. So we don't want to introduce any antigens it will go after or attack. So a type A host can receive type A or type O blood because neither of them contains the antigen B. Type B hosts can receive type B or type O because neither of them contains antigen A. Type AB can receive any of the blood types because type AB people do not have any antibodies at all, so they have no armies to attack the incoming blood. And type O can only receive type O because it has both antibodies and will attack any incoming erythrocytes other than type O. Now, there is another group of antigens that we're concerned with. The Rh group consists of multiple antigens named C, D, and E. But the D antigen is the most clinically significant. If a person is born with the D antigen, they are considered to be Rh positive. If not, they're Rh negative. And this is why blood types are typically given a positive or negative modifier, like type A negative or type B positive, for example. The difference between the ABO group is that anti-D antibodies are not naturally occurring in Rh negative people. They are only synthesized if an Rh negative host receives Rh positive blood, and then its immune system will start to make antibodies against this new antigen it just got exposed to. So for that reason, we tend to favor using Rh negative blood as donor blood because it has no D antigen for the person to attack, just in case the host had been exposed before and now has anti-D antibodies in their plasma. So it's basically the safest bet. One of the most common clinical applications of this is pregnancy. If someone is pregnant and they have Rh negative blood and the fetus is Rh positive, then the possibility is that the pregnant person could begin to produce anti-D antibodies because the blood between the infant and the pregnant person gets mixed. Now, this is not a concern for the fetus, but if that mother gets pregnant again with another Rh positive fetus, the mother's new anti-D antibodies will attack the fetal erythrocytes. And that's really bad for that fetus. So what happens is, often Rh negative pregnant people will be given an injection of a drug called Rogam, which inhibits the synthesizing of anti-D antibodies to avoid this dangerous scenario. All right, so now the biggest question here is probably, well, why doesn't the donor blood attack the host blood, right? So if we're giving someone who's type A some type O blood, well, type O blood has anti-A antibodies. So why isn't this donor blood attacking the host blood? And the reason is mostly that when you give someone blood, you're usually giving them the cellular component and not so much blood plasma. So the antibodies have been removed because the antibodies are in the plasma. And even if there is some plasma in there, the antibodies are not in high enough concentration to really do any damage. You really only have to worry about the host blood attacking the donor blood. You'd never have to really worry about it the other way around when you consider these clinical scenarios. All right, so that's a good introduction to the circulatory system and also some talk about blood composition and erythrocytes. So in our next episode, we'll get into leukocytes and we'll move forward with whatever we can get to in the time we have to talk as much about blood as we can. So I'm really glad that we have moved on to the circulatory system. I hope that this helps you get your B or better in A&P. And I want to thank everyone involved in helping out. And I also want to thank Liam from Louisville, Kentucky for sending me your question. I hope that I answered your question well enough. And don't forget, I'll put that website on the show notes so that you can look it up yourself and take a look at what I looked up when I saw that question. Once again, thank you so much for listening. I really do appreciate it. Don't be afraid to reach out to me. I will try to get your question on here, or if you have other questions, I will just try to answer them as much as I can. Good luck on your next A&P exam. I'll talk to you next time. Anatomy and Physiology Bit by Bit is a production of Minus 55 Media. Please take the time to rate the podcast, and don't forget to check out my YouTube channel, Student Help for AP. That's Student Help, the number 4, AP. There's a whole lot of tutor videos on there that I think you're going to find helpful. Special thanks to my family, Bucks County Community College, and McGraw-Hill Education, where you can find Anatomy and Physiology Digital Suite, my low-cost, tutor video-based digital learning solution for anatomy and physiology, already being used at several colleges and universities.
Your kids could get free or low-cost health coverage from Medicaid or CHIP. Even if you've applied before, they may be eligible now. Kids up to age 19 are covered for checkups, vaccines, dentist visits, hospital care, and more. And if they already have Medicaid or CHIP, remember to renew every year. Visit insurekidsnow.gov or call 877-KIDS-NOW. Paid for by the U.S. Department of Health and Human Services.